All right. Good morning, everybody, or good afternoon, evening, wherever you are. Thanks for joining us for already field day number 12 of the Agricultural Genomes to Phenomes
program
initiative. If you want to mute if you're on, please. So the Agriculture Genomes to Phenome initiative funded by USDA NIFA, and we're very grateful for their support of all these activities. My name is Jack Deckers. I'm a faculty member in animal science, animal breeding and genetics at Iowa State University, and I'm one of the co-PIs of AG2PI. And we have also Nicole Scott, who's the program specialist for the project and does all the coordination for these field days. So thanks to Nicole for that. And Eddie, who is involved in the a lot of the computational side of what we're doing and the website. So today we are going to have a field day, which we called an industry showcase. So of course, industry is an important partner and a stakeholder of AG2PI. I mean, what AG2PI is very much focused on academic research, but we can't do it without industry. Industry being implementers of the things we're developing, but also helping us develop tools and systems that can help us conduct the research that we need to do for AG2PI. So they're a very important partner. And so we wanted to give them industry an opportunity to feature their work and talk about things that could be helpful to us as a research community. So we had an open call for proposals of industry stakeholders who were interested in presenting in this field day, and the two that we are going to hear from came forward. Since then, we've heard from some other industry partners that were interested, but we didn't have an opportunity to include them in this field day. So we'll most likely fairly soon have another call for proposals from industry for a similar field day, industry showcase field day. So keep an eye out for that. Now, the purpose of this showcase, industry showcase field day is not to advertise the specific industry partners and the products that you're going to hear from. And the fact that these companies have been given the opportunity to speak at this field day should not be seen as an endorsement of these companies and their products by AG2PI. And us as members of AG2PI, the co-PIs and the PIs, we don't directly benefit or directly or indirectly benefit from giving them this opportunity. The purpose here is really for all of us to learn about new technologies that are being developed or have been developed in industry that could be applied to genomics research, and that's the purpose of these field days. So today we're going to learn about two companies and the systems that they've developed for automated phenotyping in plants. And the first one is a company called Plant-DiTech out of Israel. And they're talking about a functional plant. Well, Plant-DiTech is a functional plant phenotyping company, and we're going to specifically hear about the methodology they've developed to screen tomato genotypes with a product called PlantArray. And Katrin Jakob, who's located in Germany and leading the US business development for Plant-DiTech, is going to present that to us. So Katrin.
All right, thank you so much, Jack, for the opportunity. Am I loud enough? Everybody can hear me?
Yes.
Thank you, and I'm going to share my screen. I'm excited about the opportunity today, and I hope everybody can see it, to present our high-throughput phenotyping technology. I'm actually in California. I'm originally from Germany. As Jack mentioned, the company is headquartered in Israel. And our technology that I would like to introduce today is seen here in an overview, and I hope to convey in my discussion today a few very important points that our technology can actually do. As you see here in the overview, we are high-throughput phenotyping technology, which can first very precisely control your experimental conditions, particularly drought-stress. We have a very high resolution in our screens, but we can also scale. You can test many different plants and different experimental conditions and scenarios in one single setup. We have instant data analytics, and so you can very quickly screen in your cycles. And this is, I think, very quickly and precisely. And this is important for the breeding community, I think, and there are many other applications too, because when the molecular breeding has been very much advanced recently, but we're still having quite some bottlenecks in phenotyping, and I'll go into this in a later slide. So what I really want to take you home, we can precisely control your experimental setup, we can precisely measure, we can do it at a high resolution and at large scale, and we measure simultaneously and continuously. We all know that the climate is changing, so there is a large need to adapt our crop production to the changing climate conditions and breed for climate resilient crops. And we are quite aware of that. It has been a challenge because, especially for drought resistance, it's a multi-clonal, a multi-gene trait, and heavily depending on the environmental changes in the field or in the greenhouse. And water use efficiency is a very important parameter if we would like to screen for drought. And there are many different definitions out there, but it is in general the biomass produced for the amount of water that has been used. And as I mentioned, molecular breeding has made quite some advantages, mainly because we can sequence now very quickly. But again, there is a huge bottleneck still. How do we precisely measure on the one side the plant response in the genotype by environment scenario, and on the other side, how we precisely set up our stress condition and make sure there is a uniformity in the stress condition, and that it has been a challenge. And so we have developed a technology which describes the plant flow, the water uptake, and the water release. So we can actually describe the plant water balance precisely, which allows then the breeder to much better go back to the genomics, because you have precise data. And you can either, using our technology, when you do a forward screen, so you can go from your first pre-breeding developments into the greenhouse with our technology, do our test, screen quickly because you get instant data, and then take the phenotypes that you have been able to describe very well with our parameter that we can measure, and then select those genotypes that have the best conditions, with the best results, and take them into the field. What you can also do, if you screen in the field and you see certain phenotypes, you can go back into the greenhouse, use our method, and look more closely at what the plant is doing and what are the characteristics and the reaction of the plant to your experimental condition, particularly drought. And you can then go back and you can adapt your models, your genetic models, but also, as we have made advances in phenotyping technology in general, with remote sensing, you can even there use our technology to have better data for your models also. So I will describe the technology and how it came about, and then I'll give examples on tomato for drought-stress experiment, on quinoa for a salinity stress experiment, and also show you that you can actually, with our platform, do stress combinations. And I mentioned how it came about to develop this technology. So phenotyping challenges in general, it's very hard to capture the genotype by environment interaction, especially for the plant-water balance. Water use efficiency, as I mentioned, is affected by many different parameters, from how the root takes up the water, how it gets transpired, how the atmospheric conditions are. There are many other factors, and it's very hard to describe. Stress response is very complex, and there is a lack of methods that directly measure parameters. It's also hard in stress conditions. What do we measure? When do we measure? How often do we measure? And it's also hard to non-destructively describe patterns of root and shoot growth. Can we repeat those experiments? Very important. We want to, of course, fully automate. We want to have very high resolution, but we also want to scale. We want to test many, many plants or genotypes in one single experiment, as well as different treatments, ideally. How do we know the time for the stress? When do we apply the stress? What's the intensity of the stress? Very important also, especially for drought experiments, the uniformity of the stress. How do you make sure that every single plant or test plant in your experiment actually gets the same type of stress? And then the big question, we are a greenhouse platform, or at least it needs to be covered. Can we actually take the greenhouse data and do they apply to the field? And actually, I want to report this technology has actually come out of a university, and it's a combination of different research areas. There was a physiologist, Menachem Moshe Young, and he worked on aquaporins. This is how it came about, the technology, and he couldn't see a phenotype in mutants. And so he got frustrated and started thinking, how can we actually measure the plant-water balance? And he started to develop the technology. But because it's a complex process between soil again and plant physiology, he worked together with a soil scientist. So there was a combination of different research area that was able to develop this technology. We have the technology at different places also for scientists to use. There is a huge service center in Israel. Wageningen University also has the platform, and I'm going to show an experiment. So we make it available to scientists, but I really want to emphasize that came out of a university, and it's a great way to develop your technology into a company also, and make sure you look right and left to your other research areas that could be very helpful, the integration of different research areas. So what is the technology about? And you see a very simple cartoon here, but I can tell you it has taken quite some time to develop the technology, particularly also get rid of the pod effects. So it's a lucimeter-based system that you can see down here, which transfers the weight into an electrical charge. And we have developed a cover here, which our steel plate from the lucimeter is covered by a plastic cover to make sure the temperature is not affecting and warming up, heating up the steel plate. We have a special drainage container, which we need to have enough water so that during the experiment, you're not running out of water. And as well, because we irrigate every night to be able to measure the biomass. We measure the entire water flux from the roots, so the root uptake with a soil probe, and the transpiration, so what the plant is transpiring. We have a cover on the top to make sure the soil is not transpiring, so it's just the transpiration only that we measure, and we measure by weight loss during the day. And we measure also the daily biomass gain, and that is measured by measuring the difference between the day today and the day yesterday. We have automatic irrigation, and we irrigate not only in one single spot in the pot to prevent that, you have compaction and you always water on the same pot. We actually have 4 dripper outlets in our technology. But what is actually really, really important is our controller, and each single plant has its own controller, and that allows for several things. The first one is, again, you can precisely set up for each single pot what you are going to apply. Either it is a nutrient for nutrient use efficiency, either it's water for water use efficiency, and you can actually combine a combination. And also, when you have nutrients, at each pot you can apply actually a different concentration. So again, you have a wide array of treatments, and the controller for each plant also allows that you can add on as many units as you like to, and you can completely randomize. And our controller is also flexible. You can add on your own sensors. If you want to develop your own sensors, sometimes in a university environment, you are able to develop your own sensor and put it on the array. And the data from your sensor are also analyzed with our statistics. So we push our data that are getting collected or go on your local computer and then are pushed to the cloud. There you have an instant analysis, and it's an instant statistical analysis. So at any time during your experiment, you actually can go in and you can see what's happening to your plant and how your plant reacting. I did not mention yet, we also take into consideration, of course, the atmospheric conditions, because they're very important, especially vapor pressure deficit for stomatal-conductance. So with this system, we can really measure the water flow from the roots to the shoots, and we capture the phenotype by environment interaction. And again, we can precisely control what you are applying in your experiment. This is how it looks in real time. So it's not only one unit. So again, you can have as many units as you would like to. And up here again, I mentioned the controller, which again is which can precisely supply water supply. And because we measure, among other parameters that I'll go into, the transpiration, you have actually a few options when you set your Drought-Stress. And that's a very unique feature for the technology. You can set your Drought-Stress according to soil water content if you like to, but more importantly, you can set it to the plant own transpiration. So we know yesterday the plant has 300 milliliter transpired, and if you want to apply the same stress to each single plant, you can set it to 80% of the transpiration of the day before. And that allows, again, there's a very unique feature that each plant gets the similar stress. So that's the most that you can get to apply a similar stress. And here, actually, I would like to bring in my poll. Let me see if I can do this. Whoops. Do I have to choose? No, Nicole, I need your help. See, I only see the one that we had before. Nicole, if you don't mind, if you could load the poll, I can do it actually. If I go in polls, I see the old version. And I can only share results before we put up the poll. Okay, I hope Nicole can put it up. And the main question is actually if you, can anybody see the poll?
Yeah, we see the poll
okay,
now. The first question is, are you able to control similar drought treatments for many plants at once? Yeah.
There are several, yes, and there are, I think there are several questions in there.
Yeah, 3 questions.
Yes.
Yeah, so if everybody wants to take some time to fill that out and submit it.
Yes, that would be great. I'll leave it up a little bit, and I think I can continue with my discussion. So this provides an overview again. And it provides an overview, particularly on the parameter we are measuring, which you see on the in the middle of the picture. We are able to continuously and at the same time simultaneously for all plants in the array. Continuously means like every 3 minutes, measure the transpiration, we measure the daily biomass gain, we measure the water use efficiency, nutrient use efficiency, stomatal-conductance, very important parameter. I mentioned we measure also the atmospheric parameter, we measure the root water influx, and we can measure other soil components. We have a very highly customized water and fertigation setup. I mentioned that, and we have an instant data analytics. And our platform is suitable, comes in 2 sizes. for small plants like Arabidopsis up to trees. So we can you can test a wide array of plants in your experiments. And I'm going to end the poll, and I'll share the results a little bit later. And I'll go to the next slide. So with regard to the data output, this is what you would see. And you get a lot of data, again, every 3 minutes. So on top, you, as an example, you see the radiation vapor pressure deficit, and it's a waste-based system. Each line in this in the graph is practically a pot or a plant. And for a drought experiment, as an example, you start with well-watered condition, then you initiate the drought. You have a control group, which is on the top. You can see the bottom down here are the plants that have been exposed to drought, and then you have a recovery period. So you get many data that you can then use to analyze with your genetic data, for instance. And here are examples for tomato experiment. And I just give you some examples what you can actually measure on the data you can get. So on the upper left, you see 2 different tomato genotypes. Panelli is a wild type, M82 is a cultivar. The red has higher, you can see cumulative weight gain over the experiment. You can measure those. You can measure the cumulative transpiration, and the combination of those gives you the water use efficiency. And you can see green, actually in this particular case, the wild type has a larger, higher water use efficiency. And you can also see in this example down here a different soil water content, high soil water content, lower soil water content, different reaction of the wild type and the cultivar for the transpiration rate. So these are all data that you get with our array. So not only cumulative over the experiment, but you can also look at the daily data. Again, we measure continuously during the day. Just as an example, again, you see up here the radiation. You see the system weight, how I just showed it in the experiment for the 2 different genotypes, soil water content, and again, the transpiration rate and the stomatal-conductance. And this is over the course of a day for several days. Just the granularity of the data you can get. I also mentioned we measure root performance and the transpiration from the shoot for 2 genotypes that are very similar, and you can still see differences. The dark line is the transpiration, the lighter line is the shoot influx, and it changes during the day. The peaks are different also between the genotypes, and sometimes the root flux is in front of the transpiration, and sometimes it's the other way around. So you get very fine, high granular data that give you really insights in what the plant is doing according to the stress conditions that you apply. And a very other important parameter is the drought-stress point. That is the point when the plant really reduces its productivity. And you can see here between the 2 different genotypes, these are 2 cultivars that we actually use to test the sensitivity of the system. They show different drought-stress points at different soil water contents. And this is an example of our analytics, how it would look like. We calculate this with a fitted line, actually, just to give you an example how the analytics looks like. If you have breeding lines, you can then actually rank your lines according to the different parameter that you measure with our array, like biomass, transpiration, stomatal-conductance, drought resistance index, which again give you great insights in how your different lines react to drought stress, as an example. We do get the question, are the results transferable to the field? And in this particular case, we have 3 different tomato genotypes. And the field experiment was actually done first, and then we went back to the greenhouse. Blue is continuously watered, sufficient water supply. Orange is drought. And when you take the cumulative transpiration, it highly in the greenhouse, from the greenhouse data, it highly correlated with the total yield in the greenhouse. And all those data are actually that I show you, they're all published, and you can read up on them. This one has been submitted actually, the data on this one. And providing that you get, particularly for drought stress, you have different conditions in your experiment, first well watered, and then you have the drought, and your control line continue to take up water, and then you have the recovery phase. Now this enables you actually do correlations between field experiments. Again, you can actually, if you have field data, you can now correlate all those different data from the greenhouse and can compare it with the field data, and you can find correlation so that you can actually predict in the end how your field yield will look like. So this is just an example for the well-watered conditions here in the experiment. And again, the cumulative transpiration correlated with the total yield in the field, and you can actually see it's a good positive correlation, also a great negative correlation for total yield, and a cumulative transpiration again in the drought phase. Again, the data you can pull out of the system allow you to eventually predict the field data. So going from the drought experiment to a salinity experiment that has been done at the University of Wageningen. Down here, actually, you see a picture how this looks like in real again, just emphasizing again on the continuous measurements over the day. The green line you see here is no salt stress, and then the blue is medium salt stress, and red is high salt stress for two different quinoa lines. And you see the transpiration here. The highest transpiration, so transpiration when you have a control in your control line, while the salt stress did affect transpiration. And if you go to the water use efficiency, you can see that the salt stress induced actually has higher water use efficiency than the control line where there is no salt stress. So just giving you an example of what you can do. And while you just explained in the salt stress experiment, you could actually, in combination, also do a drought stress experiment. So again, because we control each single pod, you can do a combination of stresses. And this is a salinity potassium. So emphasizing also that you can do stress combinations in this particular, with this particular setup. And we know for drought resistant that I think the nutrient status of the plant has come into a discussion. So you could set up a nitrogen experiment with different concentration and then test your drought stress. Okay, let's see if I can go to the next slide. So when we developed the technology, we started off, of course, comparing your manual measurements to make sure the system provides correct data. And what you see here in this picture, and this has been independently done from us, at Wageningen University in the quinoa experiment I just showed you, they took manual measurements with a porometer and correlated those measurements with the data our system put out, and you can see it's a very high correlation of 0.95, and which made us very proud. So you do not need to go by hand anymore, and you would certainly not be able to do this continuously for all plants in the array. So rather you can actually have a data output that is continuously and at the same time again for each plant in the array. And again, you see here on the right side the picture just to show you again, the blue is the stomatal-conductance, and we have this peak in the morning the golden hour, so to say, for the plants, which then over time decreases because the radiation increases, and so does the vapor pressure deficit. All right, so as a summary, I want to emphasize again that our technology measures simultaneously, I mentioned that many times, directly, precisely, and objective. So you get really good data where you can then refine again your models, your genetic models, or your machine learning models in your remote sensing devices. We have a very high resolution per plant. We can measure non-destructively. And so this actually, and again, you see your data very quickly in your array. That saves you quite some time and resources in your research pipeline, so to say. And just to give you as a last picture again, where we would fit in, and I mentioned that in the beginning. So if you do a forward phenomics, right, you have developed your seeds, you have your you have worked with your molecular breeding technologies, marker-assisted breeding, you figured out which seeds you want to use, but now you can go into the greenhouse with our technology. You can apply precise conditions, repeatable conditions, and from there you can select the genotypes you want to take into the field. And then you might even ease up your the measurements because you kind of already know what your genotypes are doing. Or on the other side, when you have a field trial and you see certain genotypes in the field and you want to know more about them, you can take them back into the greenhouse onto our platform, and then you can dig deeper into why your plant showed those symptoms in the field. And let me actually see if I can pull up the poll again to share the results. Oops. So this is not, I have to go in here again. Let me see. Okay, yes. So sharing the poll results for everybody. So the first questions, are you able to control similar drought treatments? And so this is actually a really nice overview. As I mentioned, and it's very hard to be able to actually set the drought experiments at similar stress and also repeat the experiment. So this is something our platform or our technology certainly can help. And how many errors you introduce by measuring manually, and how many manpower do you need? So it would be much better if you could actually control automatically, and that's what we do. Similar stress condition, very easy. Only 8% said it's very easy. You do rely on repetition and statistics, so this is very interesting. Again, with our technology, you will be able to have a similar stress impact, particularly for drought. Or related field water stress simulations applicable to your research. No, so we have 35% who are not working on this. Some work on Abiotic-stress. Some have really, obviously, Lysimeter in the field. And it's almost similar to work with very small timescales and use models. So this is probably interesting for us to know if you do field experiments, because sometimes we get the question if we can actually apply it into the field. And we want to look into this in the future, how we might be able to transfer the technology also in the field. But again, you get so many data that you can actually well predict from the greenhouse data what's coming in the field. All right, so, and with this, I would like to end my presentation, and I'm happy to take questions.
Okay, thank you very much, Katrin. Very interesting, and as a person working with animals, I'm very jealous what you can do with plants, what you can do with animals. Although technologies are developing also for animals, but certainly it's a lot harder compared to plants being stationary. So yeah, so put questions in the chat, but you all can also just speak up. and ask questions. So there's some questions in the chat already. I think the first one from Paul Richter, I think you've answered the correlation of
yes,
what you measure in the greenhouse versus the field. The next one by Roland Bocco, yield is very affected when the drought happens during flowering. And in your case, do the plants, are they on the same cycle? And then the second question, how do you confirm the soil homogeneity?
So yes, are they on the same cycle? You can, not quite sure if I understand the question, but you can take the plants all the way to flowering. You can repeat it in the winter and the summer. There is a little bit of a setup, and you can apply the stress at any time point if you like to. So if you do want to apply your stress later, and please come forward if I did not answer the question well enough. So soil homogeneity is, we have a recommendation of different soils. We have a table that you can use, and you are supposed to, before you set up the experiment, to mix your soil very well so that the soil is actually homogeneous in your pot. And we train the pots every night, which also helps with setting off, that salt accumulates, etc. So we try to have it as homogeneous as possible.
Okay, thank you. Yeah. So please, the questioners want to follow up, just speak up or put things in the chat. Question from Sue Ree. Do you have plans to incorporate florimeter function to measure FV over FM? You'll have to explain to me what those are, because I'm not familiar with those terms, Catherine.
Yeah, neither do I, actually, I have to admit. So I guess something that has to do with the flower time or the flower characteristic.
Photosynthesis.
Oh, photosynthesis. So we do measure stomatal-conductance, which is kind of a, measure of the of the transpiration, which could be translated into photosynthesis activity. But you could, if you want to, you could also add an NDVI, or other sensors that would help you with this. But our main measurement for this is the stomatal-conductance.
Okay, and then a question about analysis. Do you offer machine learning software to work with the data?
Not really. So we have the you have all the data. That's practically what we provide. We do not provide the machine learning software. We're actually, I have to say, we're working on this, going more into machine learning also, and it remains to be open later on if we would provide this to the community or not. I mean, we could probably do arrangements there, but this is not, we are not anywhere close yet to. So we have started looking into this.
Okay, I have a question. So I mean, obviously plant density is quite key. So does the equipment come in different sizes, or how can you modify plant density?
So the array comes in two sizes. For 1 to 8 liter pots, 20 by 20 centimeter is the size, and the other is up to 40 liter pots, 40 by 40 centimeters. And how dense you put it together is up to you. You have seen in the pictures, you can put it up very close together. The only conditions you have to consider is that they do not really touch each other, because that would affect the weight. So make sure they are not touching each other, but otherwise you can space them out, or you could put them together. That is really up to you.
Okay. So that would affect, the fact that you can't have them touch each other. That would affect how it translates to the field, right?
Yeah. So you can see in the tomatoes they put very close together, but again, you do not want them to have be touching each other.
Yeah, I mean, I guess you could always do the manual weighing if you want to.
Well, that's what you want to actually prevent.
Well,
Yeah.
yes, but there's a lot of other things that you're measuring. So, but yeah, I think. Any other questions? Oh, there's another question.
No, the economic data are very important. There is no question. So I see the question in the chat. Do you mean that economic data are not important to explain drought? So yes, the field conditions are still different than the greenhouse conditions. You have the evaporation from the soil, you have the plant density, as it was just mentioned by Jack. So no, I mean, you still have to do your field experiments. But what we want to actually do with what we can do with our platform, that you can choose better the genotypes you want to take into the field in the first place. So if you select for drought resistance, you already want to go with genotypes that actually show, reduced transpiration by hopefully increased biomass, have better drought resistant parameter data, so that you actually don't waste time. Field experiments are very expensive, that you already can choose, promising genotypes. So this is one of the main reasons you would use our array. And there's no question, we cannot replace the field experiments, but we want to make it, as easy and effective as possible in selecting what you take to the field.
Okay, thank you. I think we'll close it off here for Catherine, but she'll stay online. So if there's questions that come up that are for her, just put them in the chat and we can come back to those later. So thank you very much, Catherine. And next, we're going to hear about multimodal UAS sensing solutions for field genomics by a company called Griffin. It's also a startup company out of university. It was interesting that Plant-DiTech got its start in the same way. This one is out of Purdue University, and Matt Bechtel is the CEO, and Evan Flatt is the Director of Solutions, and they're going to share with us that system and what they have available. So, Matt, go ahead and share your screen.
Thanks, Jack. Evan's going to start the share. Appreciate your time, appreciate the opportunity to connect with this audience. And a special shout out to Nicole for all the help in coordinations, and appreciate Catherine's work and what they're doing at DiTech. One of the things that we wanted to make sure that we set at the state up front is that this is going to be obviously a conversation around how we're approaching these multimodal sensing solutions. We're going to talk a little bit about our history and how we got to that point. But what we want to do is really have a conversation. So I want to set some things up and really get it to Evan, because he's been in your shoes as you're evaluating these sensing solutions. So I'll come back to that in a minute, but we will monitor the chat and address questions as quickly as possible, clearly at the end. We look forward to that discussion. First, again, our mission, our purpose, what we are focused on is developing premium sensing solutions for digital phenomics and research. And we like to say that we're a sensing company, not a drone company. A drone is one way to collect that data. Ground-based sensing is equally as important. Proximal sensing, as we just learned a little bit about, equally important. Yes, we use drones, but we have experience with those other sensing solutions. And then as I'll address at the end as we close, the future of digital phenomics is going to be an ecosystem of these sensors and solutions, like we saw with DiTech, like many other sensing solutions, like other remote sensing solutions, in addition to Griffin. We clearly recognize that need, and we'll talk a little bit about that today. Next slide. So where did we come from? We've talked about this in the past, but we would not be here without the support and funding and really sense of urgency created by the Department of Energy and the Terra program, led by Purdue University. The charge of how do you develop sensing platforms, advanced analytics, in this case specifically around sorghum as a feedstock for biofuels. But how do you advance that technology when it's very complex, it's really expensive, and unique to Purdue in this particular application? It's also very multidisciplinary. How do you bring together the kinds of knowledge and team and experience to really explore these solutions, to find meaningful solutions that can be applied beyond just a single research project? And that gets back to the last bullet on this particular slide, that most of you that are in academic research are probably aware of, but with ARPA and DARPA projects, you've got this congressional mandate to focus on technology to market. And that's really been our charge. How do you take this research and how do you make that, or how do you find common denominators? How do you make that robust, not only for an academic community, but also a commercial community? And that brings its own set of challenges. So again, those are some of the things that we want to narrate for you today in terms of our approach. How are we doing things? Why is that important, as opposed to, perhaps, our solution compared to others? And again, super helpful to have partners. The Department of Energy leading the project, led by Purdue from the PI perspective, with collaboration with University of Queensland. But also what's unique for this particular grant opportunity is we're working directly with collaborative partners where we have put solutions in their hands and we're getting that feedback loop from a diverse group of people. Small organizations, large organizations, those with lots of capacity, those with lots of experience, and also the other side of it. And that helps us build more robust approaches. Next slide. So as we think about that Terra work and some of the challenges around sensing, specifically remote sensing and outdoor sensing today, how do we accelerate digital phenotyping? How do we facilitate scalability? How do we go from a lab, small scale or small plot environment to very, very large experiments in the field? How do we focus on low-hanging fruit tasks that are labor intensive, like heights and counting and these kinds of things? How do we also focus on quality and accuracy and time series analyses? Often in our industries, we've applied consumer grade tools to our work, which have a lot of value, but were never designed for agriculture or our work. How do we maintain research grade quality so that we have consistent, reliable scientific results? How do we use that quality to fuel this coming machine learning and artificial intelligence, monsoon that many of you are evaluating? How do you make it repeatable? How do you make it as easy to use as possible? How do you free up breeders away from that, especially from the plant domain? How do you free up that time they're spent measuring and counting to focus on maybe more important, higher value types of activities that they don't have time for today, or maybe are inconsistent and somewhat subjective? And then finally, as you put these things together, how do you foster innovation, insight, and predictive capabilities by leveraging these multimodal systems? And that's work that we'll refer to, that you will see many of you are doing. And these are the kinds of things that we took to heart in our work. Next slide. And so as we've worked with these collaborators, not only with the grant, but our customers, academic partners, we're driving that feedback loop of what is important? What are the hopes, the dreams, short-term and long-term, when it comes to phenomics? What are you trying to look at? Things like plant height, obviously yield, we just talked about, silking, flowering times, predictive analytics, disease. In this particular case, this is just a small list of things from a few people for one crop. And what you'll notice about these is it's not hard to make the list. There's about 50 of us in the session right now. I'm sure we can all come up with 10 traits that we would want in both the animal and plant space. So it's a long list. So a couple of things for us that have emerged. One, obviously there's a lot of diversity. There's a lot of different needs and research objectives. So how do we take a step back to find, well, how do we best serve a broader base under that technology to market mandate that drives value for multiple people? And so this isn't research focused. I think if you click next, Evan, we're going to focus on kind of the system, the setup, the approaches, but I would highly recommend, we were talking about the AG2PI sessions that are recorded. The Purdue team did work on this in the spring. So I refer you back to that session. It's a great deep dive into some of the explicit plant science applications that are happening. And I'll refer you back to those. Next slide. So what are those common themes? Going back to that next slide, it's a diverse group. Every one of you has different objectives. So how do we bring that power to the most good? And so what we have focused on is really three things. One, how do we improve scalability? How do we take the lab to the field? So how do we bridge the controlled environment with high quality research in the field that's reliable? Two, how do we make it easy to use? They're still research tools, they're not toys, but how do you streamline that? How do you make that one person operation? How do you also make it reliable enough that you know that if the right people at the right time with the right maturity level, you can go get that information that you need? And 3, at the end of the day, you have to deliver quality, precision, and repeatability. So a lot of people are asking us about what's your analytics pipeline and what are you doing? And yes, we're evaluating those things internally, but you have to be able to deliver consistent research grade systems across time and space if you really want to advance the phenomics frontier across multiple states, across multiple regions, or across the globe. Next slide. So that said, we wanted to ask a quick poll question, and I'm going to go ahead and launch that now and then just kind of narrate real quickly. Let's relaunch that. So that should come up on your screen now. There's 3 questions. One, are you using remote sensing in your research workflow? So obviously this is outdoors and remote sensing domain, and there's multiple choices. So please check all that apply. Number 2, what do you see as the most significant opportunities for remote sensing in plant breeding specifically? But obviously there are applications for foraging, grazing, and other animal agriculture activities as well. And lastly, what do you see as the most pressing hesitation or barrier to adoption to research grade systems? And we ask these questions for a couple of reasons. One, as I look through these results and Evan is talking, clearly we'll make sure that we address some of these. But 2, obviously remote sensing and sensing of all kinds is really important to the AG2PI initiative. So this feedback will get us a better understanding of kind of what your background is and kind of where you're thinking. So I'm going to leave that up for a few minutes before we end that. And then what I'm going to do is kick it off and turn it over to Evan. So Evan Flatt is our Director of Solutions. And we know that many of you are either A, already flying kinds of systems, B, building your own systems or integrating different approaches in those technologies, C, searching for a solution so that you can embark upon that research, or lastly, you're still on the fringes and you're thinking out what is that value proposition. Evan has seen this both as a student, but also as a staff member facilitating this work at Purdue University. So I wanted to turn it to him because he can kind of narrate the lessons learned, the approaches we take. I'll monitor the questions. And then again, please, at the end, when you have questions about other remote sensing approaches, other sensors, other things, we want this to be an open kind of learning opportunity, not necessarily just focused on our particular approach. So Evan, I'll turn it to you.
Okay, thanks, Matt. So I'm just going to address sort of quickly here some of the things that Matt talked about. And I really want to hit on here is just the sort of the complex nature of this high throughput phenotyping that we're all sort of working towards. So getting this accurate research quality remote sensing data in our experience hasn't been so simple in recent years. There's a lot of complex sensor operations. We have calibrations to worry about, ground control for alignment of data, and then other things that we can't really control, like weather variability, and then also reliability of platforms is a concern as well. But we are seeing that this technology is available, but it's just sort of that we need things to be sort of streamlined and standardized in a way that's applicable for high throughput phenotyping. And there are some commercial solutions that attempt to meet these needs, but often what we see is those aren't really at the level that's required for this agricultural research. So I'm going to talk a little bit of some of the tools that we've developed to sort of meet these needs. So we're taking some off-the-shelf components, as Matt mentioned, some things available in the commercial space and sort of adapting them for use in an agricultural standpoint. And what that means is really multimodal UAV-based platforms. So we're flying multiple sensors and then a sort of streamlined data processing approach to help that. So I've got here sort of a simplified layout of a remote sensing workflow in this space. So we go from taking your research objectives, which is really the input to all of these decisions, acquiring sensors, doing the integration and testing of the sensors, integrating that with some sort of platform that flies over the field to collect the data, calibrating that system, checking the quality, collecting the data, again, quality checks on the data collection and the data processing outputs. And then once it gets through all that, you end up with a final data product, which then fuels your analysis pipeline. And again, on the data analysis side, that of course will be dependent on each user's needs. So where Griffin has really looked is in this highlighted area. We want to try to streamline this process as much as possible. So that's what I'll be sort of addressing. And to that point, I'm going to talk first about our hardware. So like we mentioned before, we're talking about multimodal remote sensing platforms. And our sort of standard configuration has been Wiener hyperspectral sensors, 3D LiDAR sensors, high resolution RGB. And these multimodal systems are really key in using time efficiently in the field, because everybody knows that's a labor intensive task to go out in the field and collect this data. But it's also important to have these quantitative measurements at small scale, such as the plot or row level, as much as possible. But at the same time, we're trying to simplify those operations as much as possible. So particularly for the LiDAR and RGB, it's a very simple operation with this system. And that allows somebody, a single person in the field, to collect this data without a large degree of training. But some other considerations that we need to take into effect are what we call sensor sovereignty, which is the ability of individual sensors on the platform to operate independently while still maintaining an adaptable design and having an objective calibration that produces accurate and stable results. So I'm going to dive in a little bit on our sort of philosophy on the sensor integration. So what we've done, as Matt mentioned, is taken off-the-shelf sensors and then done our custom integration for multi-sensor control, storage of the data, and having a user interface for the user. So we have an onboard processor that's sort of controlling this data collection process. And one of the things we've built into the system is what we call being platform agnostic. So any UAV system, or any vehicle for that matter, that could carry the weight of this sensor system and provide the power, we could attach this sensor system and collect that data. So that's just to say that we're not really reliant on the UAV system for any navigation or processing aspects of the data. We're sort of separated from that aspect to allow for that flexibility across multiple platforms. And as I sort of touched on in the previous slide, we don't really have any sort of inter-sensor dependencies. So our solutions can be customized with different sensors without any impact of one particular sensor on another. So each of the sensors is handled independently as far as its data storage and operation. And that allows you to account for any sort of sensor upgrades. For example, if an RGB camera upgrades, something else becomes available, it would be a very simple process to swap out that sensor, or a hyperspectral sensor upgrade, and so on. Those are very simple operations. So there's also been some design considerations there in allowing for the actual physical design of the system, allowing you to remove one sensor without disturbing the calibrations for the other sensors. And again, all this to say, just we want to allow for as much flexibility as possible in the hardware operation, and all that to meet individual customer needs. So this is culminated in our UAV platform that you see here. Again, we're platform agnostic, so the UAV is not really a key component of the system, but we are using the DJI M600 as a standard platform just because it's relatively easy for new pilots to learn how to fly it, and it's been very reliable for us. And on the right, you see a picture taken from the bottom of the UAV, so you can see sort of the sensor perspective. And just very quickly, this is a little more detailed layout of that configuration with the exact sensor specifications and the supporting equipment that comes along with that package. And I really want to talk about, this is sort of the key thing here, getting back to that workflow that I mentioned earlier. After you've done the sensor integration and you have sort of the hardware aspect of your system figured out, we need to calibrate that system and then process or orthorectify that data to do our time series analysis and the multimodal sensor analysis. So looking at this figure, the key thing to take away here is we have a high accuracy GNSS-INS system that gives us a trajectory for the sensors. And we have each of our 3 sensor types attached on that same platform. And we need to accurately get a trajectory for this entire system. So that's given to us by the GNSS-INS system. And that's a post-process trajectory, which relies on either publicly available base station data from a CORS network or a user's base station sitting in the field. So once we have this accurate trajectory from the GNSS-INS system, we then need to define the offsets and bore side angles between each of the sensors relative to that IMU. And this is something that's really one of the key outcomes from our partnership with the civil engineers at Purdue. And it's been integral to the, like I mentioned, the multi-sensor data alignment and the extraction of the multi-date data for your field. So I'll talk a little bit about sort of the process in which we calculate these precise parameters for the sensor orientations relative to the trajectory. And then I also just want to hit on, this is direct georeferencing. So this eliminates our need for ground control in the field, which we know historically has been a big headache for field operations, particularly in an agricultural setting where you have field implements. So you're not easily able to set up targets permanently in a field without them being hit by agricultural equipment, damaged by the weather, and so on. So a little more detail on our system calibration. This is the process of how we mathematically solve that relationship between the IMU and each of the sensors. And this uses the actual imagery or the data coming from the sensor itself, because we can't accurately measure these parameters physically on the system. So in the case of RGB and frame cameras, we can also calculate internal characteristics of the camera, such as the focal length and distortion. And then on the processing, we can actually do the bore sight, those bore sight values calculated each time we do a data collection flight. But there's also different strategies for different sensor types. So in the LiDAR data, we're dealing with 3D point cloud data. So we use a sort of a geometry-based calibration process there, where we extract geometric features from a data set from multiple orientations, and then mathematically align each of those tracks together to optimize the bore sight results to give you a aligned point cloud. And I'll show some examples of that. And then the push broom hyperspectral imaging, it's more of a traditional GCP based approach where we're using a collinearity equation to align each of the images with a known coordinate system, and then using those values for future processing. And another thing I want to mention here is just that this is a calibration process that needs to be done recurrently. So we recommend like once per year for the geometric calibration, just to make sure that any corrections will be done, and also just analyzing the stability of the calibration. So making sure that things aren't happening in the field that are damaging the system. And this is just an illustration of what a flying trajectory might look like for one of these calibration processes. And then again, some examples on LiDAR calibration here. So in the red points, this is a 3D point cloud from the LiDAR data. We can see multiple repetitions of this A-frame shape target repeated from multiple tracks when the sensor looks at the object in different orientations. And then the green points show after our calibration process how these points are all aligned on the same profile. And generally from the LiDAR data, we can expect a 2 to 5 centimeter RMSE for known ground coordinates. In the Wiener push broom calibrations, we can see this is what a sort of uncalibrated look at this the imagery would look like. You can see that adjacent flight lines or imagery do not align very well. So after our calibration process, we can see a good alignment between the individual flying tracks as well as the alignment with other sensors. And typically here we expect a 1 to 4 pixel RMSE. And just a note on how these images look, these are raw, uncalibrated 3-band representations of the hyperspectral data. So they've not been calibrated. So that's why you see some color differences in different imagery here. So after we've calibrated we've done the sensor integration, we've calibrated the system and we're ready to collect some data. This is what a typical flight might look like. So given a 45-meter altitude and a 4.5-meter flying speed, we can expect roughly 10 acres and a roughly 20-minute flight time. And this will give us, sub-centimeter resolution in the RGB data, about 4-centimeter resolution in the hyperspectral, and about 1,000 points per square meter in the LiDAR data. And again, like most things in remote sensing, this is a game of trade-offs, and it really depends on each of the user's objectives. So trading off resolution for your coverage area, thinking about flying speed relative to your signal to noise ratio in the hyperspectral imagery, as well as the point density in the LiDAR data. So these parameters can be tweaked for individual users' needs. And again, on more of the operation side, we've done quite a bit of work to streamline that because we know that the time commitment in the field is key, and it's important to hit some of these stages of the plant growth and collect that data, as well as working around weather. So just some of the things that are important to think about there. For example, on the hyperspectral data, the reflectance target deployment and maintenance, so you can calibrate the data. That's important, and we've developed these targets you see here, which allow you to quickly deploy these reflectance panels, which we found has been sort of a pain historically to have these calibrated reflectance panels set up in the field and also kept safe from abuse, and try to keep them consistent. But also other things to consider, such as system logging. So just recording what's going on with the system while it's collecting data and while you're in the field, being able to record events that happen in the field and environmental conditions, checking the data, when you're in the field, making sure the raw data you've collected is what you expect, and then automating that organization process. Logging all the parameters of that mission is important, as well as the actual UAV flight planning as well. We tend not to, focus so much on the UAV. It's more of a platform to carry our sensor system, but the planning there is important to the data quality, and we've done quite a bit of work to test and validate the different UAV flight planning softwares that are available and evaluate their pros and cons in different applications. And again, those sort of uses will be dependent for different users and their needs. So I'll talk a little bit here about our processing software. This is meant to be sort of a quick overview to show we want to try to minimize user interaction with the software and provide a consistent output once you've collected your data. We're using industry standard formats and a consistent data organization structure, but also allowing batch processing. So having a pipeline where you can bundle up your data into a consistent organization structure, feed that into a pipeline where the parameters will be consistent as you're processing this data over time, but also allowing the user to understand really what's going on behind the scenes in the software and tweak the settings that are important for research needs. So I'll sort of walk through the workflow here and step through a couple of screenshots. So the first thing, which I sort of touched on, is the raw data bundling. So this is taking the raw data as it comes off the sensor, bundling it up in a consistent format, and then feeding that into the pipeline. So we'll point to all the raw data on this page, point to a calibration file, and what we call processing extent, which allows you to define sort of the geospatial area you want to actually visualize the data in. We will define a pipeline to be used for that, to process that data. So we can set up different parameters for LiDAR, hyperspectral, RGB, etc. Define that pipeline and then point data to that pipeline, which then it's submitted. And at this step, this is where we would do the target pixel selection for the hyperspectral reflectance calibration. So that's included here as well. And then ultimately, you just hit run, and then the software runs and notifies you when that data project is complete. You can submit multiple jobs to that pipeline, so they go into a queue, and once job 1 is complete, it moves on to job 2, and so on. So as an output to this this data processing, these are sort of the core data products from each of the sensor types that we would expect. So for LiDAR, we're looking at 3D point clouds in again, a standard class format. And this is geospatial XYZ coordinate information, which we can also colorize from the RGB imagery, as well as give an intensity value for each point, each discrete point, which again is at a nominal flying altitude and speed is about 1,000 points per square meter. We can also output rasterized elevation data, as well as bare earth terrain models from that. And some of the applications that this data fuels, we're looking at plot and row segment level plant height, volume estimation, statistics on the canopy, sort of the canopy structure and the distribution of the points within the canopy, as well as penetration indices, and looking at some other structure-based features in the future. So we can just see some examples of different visualizations of the plant height here. So this is a 3D point cloud on the top view of the field, and also the just an average plot level row height, or sorry, plant height measurement. This is just a quick animation of what just a small 2-row segment extraction from that LiDAR data looks like. So you can start to see some of the actual structure of the crop here, which is pretty interesting, and just get a very accurate measurement for the height. And this is taken from, again, a nominal altitude of 45 meters on one of our standard LiDAR systems. So getting into the hyperspectral data, looking at the veneer, here we're talking about the core data product being a hyperspectral reflectance orthomosaic. Some of the applications for this, again, this is at the plot or row segment level. We can look at false color imagery, narrow band vegetation indices, which I've got here. And these are really, this is a lot of data here that can be used to fuel machine learning and some of the phenotypic prediction models. And then lastly, I'll talk about the RGB. So again, the core product here is an RGB 3-band mosaic at a really high resolution, so sub-centimeter resolution. And you can also generate sparse point clouds from that as well. And the applications here are going to be at a smaller scale, so plant or leaf level even, depending on the flying altitude. And you can do things like plant counting. You can start to look at leaf segmentation and panicle and tassel segmentation from this high resolution data. So this is an example here of one of our plant counting outputs. And lastly, this is just a quick video looking at some of the raw RGB imagery, so you can just see the level of detail that's expected in this imagery. So very high resolution, which allows you to look at a lot of interesting characteristics of each plant. So hopefully I didn't run through that too quickly, but that's basically what I wanted to show, and I'll be happy to answer any questions, Matt and I.
Yeah, in summary, to kind of do a couple things. One, I'm going to share the results of the poll, so you should see that coming up here in a minute. And again, so we wanted to give you an overview of some of the common denominators that we found in our work, approaches that we're taking to achieve those set expectations in terms of taking the lab to field, trying to improve scalability, trying to improve usability, and trying to provide a platform. You go back to that slide with the maze on it. How do you provide a multimodal solution that's accurate for time series, has the resolution that could be necessary for the physical type of traits like height or biomass and those kinds of things, or spectral or chemical information, in addition to those machine learning applications, so that you can serve the needs for a broad base of customers. We do not pretend to be the only solution or approach. There's going to be an entire ecosystem of these. Even with our platform, for a lot of organizations, a small subset are going to be at this level of quality for lab work, whereas others, again, back to the research objective, are going to be specifically designed to be doing other things. So what we've tried to do is build in as much agility and reliability in that as we can. Over the last couple of years, we alone have flown over 500 missions. So we have learned a lot about what works in the field, what doesn't, how do you streamline, what's reliable and what's not. And so, again, we really appreciate your time today. We're going to turn it over to questions. Obviously, if you want to get into the specifics of the platform and capabilities, please email us, send us a note. We can send you more information. But for now, we really want to open it up to any questions you might have in general about the application, our work, or other applications. From the poll, you can tell there's quite a bit of diversity in experience and applications here. I know that I did answer one question already. It looks like there's a new question. Does your software have APIs or integrations to easily connect with other systems? What data is available through the API, et cetera? So we currently do not have an API, but we do focus on exposing some of that command line work for some of the research type of activities. But the API is something that we're talking about doing so that we can easily flow into enterprise workflows. So that's something that we're evaluating now. The two most common requests so far have been one, command line, and two, Linux for that community. And so that's something that we're developing this fall. Other questions?
Hey, thanks, Matt and Evan. Very, very interesting. Yeah, so please put questions in the chat or just speak up. The floor is open. As we're waiting for that, I can definitely see the animal science applications of this technology. And have you worked with animal research projects with this technology?
Only a little bit. So for example, I know Purdue has been conducting research around forage and sorghum and trying to use remote sensing applications to monitor grazing patterns and impacts on the plant over time. So we have definitely supported a couple of those flights, not the analysis as well. But we are continuing to see increasing applications they tend to be primarily in grazing and forage, perhaps a bit more indirect. What's also really interesting is the ability to leverage other sensors such as thermal, so that you can start to look for animals at night, a little bit easier to pick them out depending on the breed, starting to look at impacts on soil erosion or overgrazing and those kinds of things. But that's about the limit. Obviously, there's exciting things that are happening in animal agriculture in proximal sensing with facial recognition and pattern recognition. And I think over time, those will continue to merge.
Yeah, and I can see applications in wildlife or just evaluating or monitoring natural environments.
Exactly. And again, to Evan's point about the flexibility, that was one of the early lessons in that, a lot of people that we've worked with have had experiences where they've really needed some sort of imaging platform, but when they have acquired one, it's really limited them in terms of their ability to be agile with either the addition of sensors or upgrading, and so that's why in that flow chart we always start with what are the objectives. Very often I'm famous for when you're a hammer, the whole world is a nail. A lot of times we take these systems and then we try to apply them to our research. And what we have found is most researchers really need a bit more customization to that, and which is why we've tried to build in that agility.
Okay. Questions from the floor? Anybody? So how big of an area? I mean, Evan, you mentioned 10 acres. Of course, it depends on the question that or the research objectives, but what's the largest area that you've worked with?
So I believe it's, Matt, you can correct me, but somewhere between 30 and 40 acres for a single mission. So a site that we're hitting multiple times. And so those sort of flights, those typically require, 3 different flights. But that's something we built into our software actually to organize multiple mission flights. So all of your data for that field is organized in a consistent way, and it's broken up into those individual missions. And another thing I guess I'll mention there is it's important when you're looking at those multiple flights to think about the weather change over that time. So particularly with the hyperspectral imaging, being aware of the sun angle and the cloud conditions when you're in the field for a long time like that. So that can be a bit of a challenge.
Yeah, and just a couple of points on that. I mean, one, as Evan pointed out, we are obsessing about sun angle, and so due to that particular sensor, we watch that, and again, time series analysis, doing those flights pre-emergence in May, for example, in Indiana or Illinois, as opposed to late season dry down, stay green type of activities. That sun angle changes dramatically over the season, which means your window changes. So that gets back to that, high quality research grade and the discipline that it takes to do that. How do we streamline that? It also comes down to choice of sensors. So for example, our standard VLP-16 sensor that we use is good for about 100 meters of flight altitude, which for a lot of plot-based work is fine. But if you wanted to fly higher and then therefore cover more scale, but still maintain a high level of quality for LiDAR, then that would require an update in the VLP systems to get that. So that's why we always dive deeply into kind of what are the immediate objectives, what other uses might you have. A lot of our collaborators and customers, especially in the university setting, will take that same platform and then share it with forestry, for example, or environmental resources, and they'll have very different objectives. So we always try to get a picture of what are those objectives so that you make sure that the investment you have gives you the biggest return on your investment.
Yeah, no, that makes sense. There's a question in the chat from Jeff Boutain. Question is whether you can share a simple example of results of photos or images from the sorghum and grazing project, Matt, that you talked about, and for example, pre and post tassel. I don't know if you have them at hand, but
so what I would do on that is I would refer you back to earlier in the slides, and I'm sure that this recording will be available, obviously, but the field day number 5, which Purdue offered, I think they talked, Dr. Teinster talked about Shelby Gruss's work. Shelby's a PhD student, and they show some examples in there. So I think maybe I would start there, and then if not, you could reach directly to Dr. Teinster for more information.
Mhm. Yeah, I remember him talking about that. Yes. Yeah. Other questions? Anybody? Or either for the Purdue group, the Griffin, or for Katrin? Oh, there's a. Okay, so Jeff says he missed the fifth field day, but we'll go back. So just out of interest, what does the Griffin stand for?
Griffin does not stand for anything, but if
Okay.
you go back to the original Purdue seal, the original Purdue seal has a griffin on it, and the original seal at the university talks about art, science, and technology. And when you think about remote sensing and integration, we really are combining the spirit of all three of those. And so it is a little bit of a nod back to our Purdue roots.
Okay, interesting. Yeah. So I was before I joined Iowa State, I was at the University of Guelph in Canada, and they're the Griffins.
Yes.
Their mascot is the Griffin. So, all right. Well, thank you very much. Nicole, can you put up the final slide as we wrap this up? So yeah, I want to thank Katrin and Matt and sorry, Evan. Sorry, Matt and Evan very much for their presentations today, and shared a lot of very interesting information. And not only what's available, what they have available, but also the opportunities and the challenges of doing some of this work. And so feel free to reach out to them if you want more information. And it's interesting that both of them came out of university research. So that's and I think another output from potential output from AG2PI is economic development in that sense. So to wrap this up, if you have ideas for other field days, please let us know. And on AG2PI, our website, ag2pi.org, you can also sign up for mailing lists and upcoming activities. Share it with your colleagues that you think may be interested. There's a Slack channel for this field day that will continue, so you can continue the conversation. Also ask you to, as you sign off, fill out the survey on today's event, so we get the feedback on what you liked, what you didn't like, and what else you'd like to see. And then mark your calendars. The field days will be every third Wednesday of the month. Keep in mind that there's a time change between now and next month's field day. So that may change depending on where you are. There will be a training workshop on field rovers on November 12th. So please sign up for that if you're interested. And then there will be another community survey that we will release soon. So please watch your inbox for that, and hopefully you can participate in that and so we can get additional input from the community about what the needs are, because that's really one of the goals of AG2PI is not only to share, what we're all doing that we can benefit from, but also to let USDA and Congress know what are the needs to if we want to embark on wide-scale agriculture genomes to phenomes research. So with that, I'm going to close it off. So again, thanks to the speakers, thanks to Nicole and others at Iowa State for getting this all set up, and hope you enjoy the rest of the day.